Hello. Today we are dealing with another new topic, and that topic is the PDB. In my earlier lecture, I just mentioned you how to search the sequence database. Normally, the biological database, which are written in the form of the two different languages, one the nucleic acid and another one the protein, the structural database that belongs to the protein. So, in order to search any type of the structures related to any protein, this bioinformatics has provided one website, and that website is PDB. In the PDB, there are the facilities to search all the structural information regarding any protein. So we can understand this by a PowerPoint. So PDB, it is a protein data bank, and it was started in 1971, and now this PDB is celebrating golden jubilee years in 2021, and the initial data which was available in 2011, now it has reached to about many data in 2021. So basically it is a single worldwide repository for the processing and distribution of 3D biological data, and it is an archive of experimentally determined 3D structures of biological macromolecules. And initially it was established by the RCSB, that is is Research Collaborator of Structural Bioinformatics at Brookhaven National Laboratory, USA. So this data contains, or it can give you the informations about the atomic coordinates, bibliographic citations, then primary and the secondary structure informations, crystallographic structure factors, and all this data are obtained by the some of the wet lab experiments like X-ray crystallography, X-ray diffractions, then NMR, then neutron diffraction, and recently cryo-electron microscopy. So the biochemist and the molecular biologist or biotechnologist, they are doing this wet lab experiments in their lab, and accordingly, or they are submitting the details of the search proteins. So far we have studied the sequence, but any protein, when it synthesizes inside a cell, then it undergoes progressive process of folding, under the regulation of the chaperones and chaperonin proteins. These all are heat shock proteins. So once a protein is synthesized, its biological functions lies in the secondary structure tertiary structure, and quaternary structure. Primary structure, it only deals with the chain of the amino acids from N terminal to C terminals with two bonds. One is the peptide bond and another one is the disulfide bonds. But once the energy lowers, then immediately they forms a secondary structures which are decided by the two bonds which are situated on the either side of the peptide bond. So this is the peptide bond which are formed by the interaction of the carboxylic acid and the amino group, and the bond form is C over NH. But C O NH. But on this side, the another amino acids with the terminal of COH group, and also this is the another amino acid with the terminal group C over H. Okay. So these are the amino acids, two amino acids. One amino acid is this, and another one is this. So these are the peptide bonds, and on either sides, the phi and the psi bonds are there, which determines the secondary structures. The value has been plotted by the Ramchandran in a quadrant, and this value varies from 0 to minus 80, 0 to plus 180, and this determines the or decides the what will be the secondary structures like alpha, beta pleated sheets, or turn, or loop, whatever it is. So this protein undergoes a very progressive from primary to secondary to tertiary to quaternary. So how these 3D structures which are assembled monopolypeptide monopolypeptide in relations to water. So when a given polypeptide interacts with the water, then the region which are hydrophobic, they are oriented towards the inside, and the region which are hydrophilic, they exposed. So if you see that this is a this is a protein, and this protein is this region is hydrophobic, then what will happen after the foldings? This will expose, then it will goes, then this will expose, this will goes, and this will expose. So this is this regions, this hydrophilic regions, they exposed and they are inside. So this is the inside and this is the outside. Okay. So the tertiary structures is also the assembled structure. And when many polypeptide they assemble together, it form the quaternary structures. So, how to determine these quaternary structures or the 3D structures? Not only that, any protein that also interact with the certain ligand molecules, certain ions molecules. So, if you want to see the interactive sites in the 3D form, so we can go through the PDB. So, a given protein undergoes the progressive primary, secondary, tertiary, and the quaternary structures. So, let us see that how this PDB can be searched out through the various menu, submenu from the home page. So, how to access this protein through the site www.rcsb.org. But recently, this RCSB has been also modified into worldwide PDB. But here we are using this old one, that is www.rcsb.org. Either you click this home page and see how you can search any proteins. So in order to search any proteins, you have to type the name of that very proteins, or you can enter the name of the ligand or the ID of the protein. If you search, see this home page, then home page has got the so many types of the option, or you can say the menu, like deposit, then search, then visualize, analyze, download, learn more, documentations. So, I told you that the PDB purpose is that to search the data, to validate the data, to collect the data, to manage the protein. So, all this data they belong to the structural one. So, let us see that I have to search the proteins globin. You can see the protein globin, globin, globin. So once you will type then here is the oxygen binding, oxygen storage, oxygen transport. So let us just see the oxygen binding globin protein. Okay, once you will click this oxygen binding globin, then again this will come another search item. So what here you can do, आप यहाँ से आप ये जो आपके जो चार ये कोड दिए गए हैं, एक्चुअली दिस इज द identifier ID, and it consists one number and the three alphabets. So this is the heterodimeric myoglobin formed by the domain swapping. Let us see that. And here also you can see uh, many other type of the informations, like you see this structure was submitted by the Lin YW Nago in 2014, and it was released. Of course, it is the date, and the structure was obtained by the wet lab experiment, that is the X-ray diffractions with dimension 2 angstrom, and the organism is the Eucus caballus. It means horse, and the name of the protein is the myoglobin, and the unique ligand is the hem. Okay, so this is the aqueous, but you want to search the search by the name of the organism. So here you can this is the equus cabulus is there. You just clear all these things and you will just you can write in the search box. Okay, you can write in the search box. But here for the beginners, just you click this one or you copy this one and you paste this ID and you can get the result. So many lateral methods are here. So one method is that you just copy it and search the protein structures by the ID, or you can directly kill directly आप इसको click कीजिए and you can get the result. So here I am directly clicking this structures ID 3WYO. Once you will click, you can get the result. So this result has come in this one. So this page is only giving you the information about the one the single filtered ID 3WYO, and this 3WYO it belongs to the heterodimeric myoglobin formed by the domain swapping, and it is the oxygen binding domain belongs to the organ equus caballus. Okay, and this protein was expressed in the E. coli expression vector में. And now the method is of course the X-ray diffractions. These all are the X-ray diffraction data. But if you want to just have the clear view of the 3D structures, then you click the structures, and you can get the another new page with the structure of this 3WYO, which is an oxygen binding globin domain or protein. Only the regions which binds with oxygens only you can see here. So let us see that how many amino acids, what are the amino acids they are interacting with the ligand, that is heme. So now you can see this is the protein sequence. Okay, and after that this is the structures. But if you want to see the details of the structures, then what happens, this is in the JavaScript, you would have to change into the NGL WEG WebGL. So this will give you the display of the 3D structures, and just you open the full screen and you can visualize this one. So let us just the full screen. Okay. So this is the full screen structures of the 3YOO, Y3WYO, and these are the you can say now you see how this alpha helix and that this is the turn, and then this is the coil regions. So you can see what are the this polypeptide it consists of. There is also some of the facilities that this is the cartoon structures. If you want to see the another structural form, you can have the backbone like that. This is the backbone structures. Then if we can have the space fill structures, so here you just see wherever you click, you can get the amino acids. Okay, so you can just rotate it. You can rotate it, but here the ligand. This is the where this ligand is there. Now you see this is the ligand one. Okay, this is the ligand. Okay, let me see. So this is the you cannot see here. Okay, so this is the this one is the uh, ligand. Okay, this is the ligand forms, or you can see. The another structures, for examples, the let it be cartoon structure. Okay, सो कार्टून स्ट्रक्चर्स में आपको ये पूरा लिगैंड आपको दिख जाएगा, ठीक है। ये लिगैंड कहाँ पे लिगैंड है ये। सो if you want to see that ki how this protein they interact with the water molecules, you click the water, then you can see that ki so many water molecules they are interacting with these very proteins. Okay, and now if you want to see the ligand, so this is the where is the ligand is okay, this is the ligand. Yeah. I can remove this water mark, then okay, this is the ligand. Now here all this color they depict the types of the amino acids. So dark region is the N terminal and the light one is the C terminal. So whatever the color is there, the darkest one is the N terminal and is the C terminal. So if you want to have the structural view documentations, what are this blue one, gray one, red one, it represent. Then let's have the structural view documentation. So if you see the structural view documentations, then here you will get the certain informations. For examples, I told you that this is the uh, this is the dark one, the better, poor, or like that. It is by the density, and if you see, this is the low highlight ligand view option. Of course, these are the hydrogen or the hydrophobic bonds, metal interactions, then cation pi interaction, pi stacking interactions. So many informations you can have this one. So what are the different types of the bonds are there? And another one thing is that if you just see the 3D structures, then wherever you will just give this press, then you can get the amino acids. Okay, so let us have the this view, space field view. Okay, now see this is the atom lysine. Okay, are you able to see lysine? This is this is the lysines. This is this is threonine. This is proline. So you can have the wherever you will click, you can get the name of the amino acids that is them. So if you want to find out the how the ligands are interacting. Okay, so this is obviously this red one. Let it be spin. Okay, stop it. Then you have the uh, orthographic camera. Okay. Now, if you want to see the where the ligand, yeah, uh, yeah, this is the ligand. Yes. So, if you want to see the ligand, let me see, check where the ligand is there. Yeah, this is the ligand region. Okay. So, if you want to see the key, this ligand is interacting with which protein there. So, this is the histidine. Then here, this is the This is the leucines. So you can get that result, or you can have the structural details, structural view, and there also you can have the information. So let me see whether the what are the secondary structures present in this. Okay, so you just scroll it and you can have the, this is the alpha helix, then 310 helix, pi helix, beta strand, beta turn. These are by the residues. The red one is the HDA, HDG, IDI. So these are the single alphabets. So by hydrophobicity, if you want to see the colored from the colored from red hydrophilic to green hydrophobic. When it's red, it is the hydrophilic. When it's green, hydrophobic. It uses the experimental to determine hydrophobicity scale based on the whole reduce the free energies. ओके बाय एलिमेंट अगर आपको डिस्क्राइब करना है तो एलानीन ब्लू होगा, तो आर्जिनिन एएसएन होगा, तो दिस वन. So it is a very good to study the all the structural details of the any proteins. So you can have the practice on these things, and whatever the informations you want to search, you can go for this. I think that this is initially it is a good to visualize all the detailed structure of the any proteins, uh, and so at last we can have the result. We can have the applications of this very PDB. So this protein data bank is the central archive of experimentally solved biomolecular structure from which users can visualize the 3D structures or get the related publications, also get the related information through annotations, and uh, however, the PDB only allows the data retrieval and doesn't provide functionality for the collaboration or user feedback. So, although it is good to see this one, but some drawbacks are there. So, uh, so let me say thanks. Or if you have any suggestions, you can give the suggestions on my comment box. Okay. So we will have the things for the better explanations in the next time. Thank you very much.